Hello and welcome to part 3 of the FlowCytoScript tutorial. In this part, we're going to talk about how you load the FCS files that was exported earlier into the FlowCytoScript for processing with tSNE and UMAP. This is actually probably the most difficult part of the process. And so this is the part where you'll run into problems, and I hope that we're going to address some of the likely issues that you will run into here. And you can refer to the tutorial overview to see the types of problems that we're going to get into here. So the first thing we need to do is copy over the FlowCytoScript files. So you have a couple different aspects here. One, if you look at the script, you see that there are 3 folders in here. You have the source files, which contain the actual scripts used for analyzing the data. And then we have the example data files for human brain analysis. And we have the example analysis output from those same files. The output contains the analysis parameter file, which is what you will need to copy and modify each time you do an analysis. We need to copy the source file once, and that is going to be in a static location. So you can see that up here, I have already copied over the source files that we need. If you want to copy over the brain data files and the brain parameter file, you can rerun that analysis and see if you get the same output as before. You should, provided you set your seed to the same number. Right, so what we need to do is copy this parameter file and modify it for our experiment. So we are going to start with our CD3 T cells here. And so we're going to create a new folder inside our analysis folder. We have a data folder, and now we're going to have an analysis or output folder. And inside that folder, I'm going to mirror the structure that I have in here. So I have a CD3 T cells file and a viable cells file. And inside the CD3 T cells, I'm going to paste that parameter file. Now, one of the things we discussed earlier when operating R is that you need to set your working directory. If you open this parameter file inside the folder where you want all of your analysis output to go, that will tell RStudio that this is your working directory. So all you have to do is make sure that you don't have RStudio or R running, and then click on this. And to verify that, we can type in get working directory. And you can see that I have this as my folder, which is precisely this. And I don't have to type it all in that long string and risk typos to get it incorrect. You can also see that I have opened whatever I had open last time, which includes a parameter file from my previous analysis. So whichever one is on top is the one that I have most recently opened, and I would want to close this. If I'm uncertain, I can close everything, close this down, and open this up again, and then it should be the only thing open. All right, great. So now we need to modify this. And we need to modify it for use on your computer. I'll show you how to modify it on mine, but there are certain aspects, such as the syntax for defining the directory location that may be different, particularly if you're working on a Mac. And so if you're working on a Mac, it may be best to ask somebody who regularly performs these analysis on a Mac, such as, say, Lydia in Leuven Lab. All right, so first off, we can just scroll through here and see what we have. Each of these green lines is a line that is there for you to understand what is going on. It does not tell the computer anything. Anytime there's a hashtag, the computer just ignores that line. So this is our parameter file. These are the data parameters. And then everything that starts without a hashtag is something that's an instruction for R. So what we need to do is define where the data are, what our conditions or groups are called based on the names of the FCS files. If we want to rename those, such as changing them from all caps to a mix of lowercase, uppercase, or something more complicated, we can do that here. And in this part, we set the parameters that are defining the flow panel. So these are the channels that are in our FCS file, and we're going to select which ones we actually want to use for the analysis. So for instance, if we've already selected based on CD3, the channel that is a marker, the fluorescent marker for CD3, we would not include here. Similarly, we don't generally include our forward scatter and side scatter parameters here, although you might if you were interested in looking at granulocytes. So for this, we're really interested in looking at which of the channels are changing in our data set that we think are relevant for our population. So that would be markers that are expressed on that cell population and not things that aren't, because things that aren't will only add noise and confusion to the analysis. Next, you can take those names as they appear in the FCS file, which is this, and change them to a name that is more biologically meaningful, which in this case is our protein. Thirdly, we have a set here to define the scaling factor, which we'll come back to later. This is one of the trickier parts, but we'll touch on that in the next segment. The other parameters down below, we won't deal with at the moment because they're more relevant for defining the outputs, how much analysis you're going to do, and how the graphics come out. What we do need to change also is where the source files are for the script, and that's here. So you can see that both for this FCS source directory and for the FCS data directory, we have a ... nomenclature, which is what Carlos was using, and I'm sure that those of you who are more familiar with computers will be able to figure out how to do this better than me. But what we want to do is tell it where our FCS data directory is. And for instance, if you aren't certain, if you've set up your structure the way I have, what you can do is say, type in your get working directory. And this is where your analysis is. So if we look at that, we can see that the first part of that is the same to get to our data and our FCS files. So I can take this, copy it, and paste it here. If I do that, exactly that, it's going to look for FCS files in this location. And there's none there because that's my analysis out compartment. But if I change that output to data, it will find these ones. So all I need to do now is change this. That's why I'm having you do a repetitive naming structure so that you have to do less work later. Every time you make a change, you will see that this turns red with an asterisk. If you want to save that changes, click here. You should only be making modifications to the file that is named parameter. If you accidentally change something in the actual script, don't save it. If you think you've made a modification, delete it and get a new fresh copy of the source code. All right, now we're going to define our working group conditions. So what we have here in CD3 T cells, we have multiple things we could look at. So we don't necessarily have to analyze every file that's in here. We could, for instance, just look at the blood and compare wild type versus EMF regular knockout. So to do that, what we could do is click in here and select this part that we're interested in. And I do this because that way you get the exact text that's in the flow file and you don't risk any typos, because if you have a typo, it won't pick it up. And we can paste that here. And then our second condition could be EMF regular knockout. And we could put that here. And then these other conditions, which we don't have, we delete and we make sure there's no final comma. So then we would have two conditions, and basically we would be comparing wild type versus M for regular knockout in the blood. And it won't pick up these other wild type or M for regular knockouts because it'll do string matching, and you'll see that there's no blood here. It can't match these. For the next part, we would want to rename our conditions to match these, and we might, in this case, only want to have a label that says wild type and M for regular knockout. And we could even write out M for regular, like that if you wanted. In general, having a shorter name is going to be better because it's going to fit better on the graphs. Right? So then we need to tell it what that corresponds to, like that. And then we would delete the rest of these. So then you would just be running the comparison between the blood and the M for regular knockout. And even though there are other files in this folder, it won't pick them up to run in the script. So you can do all sorts of different analyses of these data, just selecting from the files that are in here. And then you can run it again if you wanted to analyze each of these. So if we wanted to do then add on spleen, for instance, we can just copy this, paste it here, and then do a find replace. So we replace blood with spleen. And then we could copy this section, add a comma. And then here we would want to do all right, so then we would have 4 groups. The order that you put the groups in here defines the order that they come out in some of your analyses downstream, particularly in the bar chart showing the frequency of the clusters. So in general, it's good practice to always put your wild type before you knock out. Otherwise, think about what's the biological relevance of doing these things. If you're interested primarily in, say, lung, put lung first. If you have a control data set, put the control data set first, so on. You would need to come back and rerun the entire thing, redefining this if you put them in the wrong order. Rerunning is actually relatively quick because you'll run the tSNE from the cached file. All right, and then one final permutation. Say we wanted to look at this tag that we put in earlier, blood versus not blood. We could do that pretty easily because we could just put in blood here and not blood here, deleting this M for regular knockout. But say we wanted to have 4 groups, blood, wild type, and knockout, and not blood, wild type, and knockout. Then we have the issue that there's this PP or spleen in between the variable that defines wild type and knockout. So we would need to actually delete that in the flow file. Somebody out there may have a better way of doing this, but I'll just show you how I would do that myself, which is a bit time consuming. So if we wanted to have blood and not blood, wild type and knockout, so we would have blood, wild type, and blood, arag knockout. Then we would have, let's say, not blood, wild type, and not blood, M for regular knockout, where the not blood includes both pairs, patches, and spleen files. Then we would need to go through and click on this, click in here, and delete that. So now we have this string that it will pick up, not blood, M for regular knockout, which matches what we've typed in there. Yeah. So to revert that, I'll just return that to that. Same thing here for the wild type. For instance, we would be deleting this section, and now we'd have not blood wild type, which matches not blood wild type here. All right, let's go back and just do a comparison between the blood and the spleen, wild type, and knockout. So we have our 4 conditions. All right, so we save that. Our FC conditions are this. Our condition labels are this. And now we need to say what channels are we going to use. For this, we need to go back to FlowJo and select the channels by looking at the flow file and understanding which ones are actually relevant for our population in question. So we have T cells. So we have FOXP3, that's expressed on T cells, TBET, IRF4, Chi67, CDCR9, and so on. So we need to change all of this from the example panel here. And what we want to put in is everything in the name here up to the set of double colons. So for instance, for FOXP3, we would put in FJ-comp-APC-A, which is already here in the first one. So we can modify the second one. Okay. And now this needs to be an exact match. So if you lack a space, or if there is actually a space you see here between Alexa and Floor, which I haven't put in, it will not pick it up. And there's a space between R and F, and in the 594, you need to have that, or it's not going to detect it. All right, and then we have Alexa Floor 647. And then we have 700. BB515. BB660 -P2. BB790-P. And if you aren't certain whether something's relevant, have a look at it in your data set or ask somebody. And for clarity's sake, I recommend sticking with the same order that the parameters are listed here, so that if you miss one, you'll be able to pick it out a bit easier. 496, 563, 615. Now here, 661 is CD19, and we have excluded that, so I would not include that in the analysis. So we would skip over that and go to BUV737. All right, BV650, 711, 750. When we reach the end, of the previous list, we need to add on more. Okay. Viability, we're not using because we excluded that. So we'll skip over that to PE. So we're not going to use the CD3 because we already used that to gate to this point. Unless you're interested in levels of CD3 within the cells, for instance, distinguishing gamma delta cells based on their higher expression of CD3. See if there's a space there. There is. All right, and after the last one, no comma. Then hit save. So that's the first one. Those are the FCS channels. And you can see there's more because we're using the Aurora rather than the Symphony, which the original parameter file was for. Next, we need to type in the FCS channel labels that correspond to these. So in this panel, which was Teresa's human panel, you can see this is the structure. And we're going to have a different structure because we're using a mouse Aurora panel. And that's going to be the case that every time you have a different panel, you will need a different parameter file. The parameter file, this part, shouldn't change that much between different analyses of the same flow panel. So if you're running the same panel again, you'll probably be able to use basically the same settings, just changing these FCS conditions and the FCS directory and possibly a couple of other minor things. So this tedious aspect that we're doing right now really only matters the first time you run a particular panel. Right, so the FCS channel labels we need to define, and you can see that the structure here is that we're going to take one of these things up above, the name in FlowJo for the channel, and call it something else. And in order to have this match exactly what before we have in the FCS channels, the easiest thing is just to copy this and then take all of this and replace it. And then we're going to insert an equals marker name here before the comma. And that's going to be the case for each channel. And you may need to refer back to FlowJo to know what the marker is. And you can see up here, it doesn't like something I've done, which is that I forgot the equal sign. As a side note, if you have a marker that is absent on one of your sets, say for instance you were looking at CD25 knockouts, you probably wouldn't want to include that in your analysis, your tSNE analysis, because that's going to be gating based off of clustering your cells based on the absence of that marker, which is genetically determined. So you already know that post pre-hoc. So you'd want to be looking at, well, what does the absence of that gene have on, what effect does that have on all of my other markers, and not allowing the tSNE to separate just off of the genetic difference that you already know is there. Similarly, you might run in that situation if you were using CD45.1 and 0.2 cells. You would separate those cells first in FlowJo rather than trying to segregate them based off of 0.1 and 0.2 in the tSNE plot, which is just not an interesting analysis. All right, so that's all of our channels named. And then the final part is the scaling. The scaling we will come to as we iterate the data set through the CytoScript. So for now, to start with, I would suggest that you just copy this section, replace this here, and set all of these to 200. Not 200 in quotes, but 200. And we need the comma afterwards, which I've been accidentally deleting here. The comma separates the arguments. Now, what we're going to come back and do later is define a unique value for each of these. Because in each channel, there will be a difference in how much noise and spreading there is in the negative. And this allows us to compress that more or less for each individual channel, and thereby get a better resolution in the tSNE. Right, so let's save that. Now we also need to tell it where our source directory is. So this you will change once, and then you can copy your parameter file that where you've modified this to use in the future with that source directory modification in there, because that's always going to be true unless you start moving your scripts around, which you really shouldn't do. All right, so for me, you can see this is my location. So it's going to be D Carlos tSNE script /00 source. So it should look in this location for the source scripts. The FCS seed base, this is the seed that starts the random part of the tSNE, and as long as you keep that constant, you should be able to reproduce any tSNE that you've run with that same seed. If you are worried that you have a weird result, you can change that seed and run it again, and you'll get a different shape, and hopefully it'll look sort of similar, but just sort of rotate it around. The other stuff we will get to later. So let's save this for now, and then see if we can load in our data. So for this, we're going to go to the source files and open up the script that says analyze flow cytometry. We'll now scroll up to the top of this. And you'll see here that the first thing it says is what it is. This is for you. And then these are the first instructions for the computer. So these are going to be the libraries, the packages that it requires. You will need to install these if they're not already installed, and it should prompt you for that when you try to run it if they're not installed. So what we're going to do is go through the script and execute each set of codes that are between these hashed instructions. And we'll do that one at a time and see where you run into errors. So start here. So to execute, we're going to select that and then hold down Control and Enter. Right. So the ret text is responses from the computer, and there's no error messages here. It's just talking to us. So that's all worked. So now we're going to go to the next one. That's fine. Next set. It's going to locate your parameter file. And then here it's going to do a consistency check to make sure that you haven't put in typos, particularly in this part where we're defining the channels that you're going to use. So if you did the setup the way I told you to and copied and pasted between those sections, you should have exactly the same structure in all three places. If you've typed it in by hand, chances are, if you're like me, you'll have a typo and then you'll get an error here. So we're going to select this and no errors. All right, source functions. It's going to look for the source files inside that source folder, and it's found them. Now it's going to create some directories inside your output folder. So let's go to our output folder. Right now, we have no folders in here. Select this, and now we go back, and you can see it's created these folders as it said it would do here. Now, this is the next part, is the part that is potentially problematic. So we're going to go and read in the FCS files. So everything from here to this rbind flow data command, we want to select this one and run it. And now it's going to start thinking, and you can see it's taking a bit because there's a lot of data to run in, and this will probably take a while if you have a slower computer. So this set of warnings here that we're not seeing is warnings that aren't critical, and that refers to the transformation of the data being slightly off scale over the max, which I showed you before in FlowJo. The way it comes up, sometimes it puts the data off the axis. That should be okay. There may be a way to fix that in the future that I don't know about. All right, so this is what it looks like if it's worked. What you may see happening is that you have an offset error, in which case you'll get red warning messages here, and it won't complete, and you'll get a bunch more errors. So I think what we're going to do in a little bit is show you how to produce some of those errors so you can tell what's happening in those cases. But right now, since this has worked, we're going to proceed. And if it's worked for you, just go ahead and proceed. So the next parts we're going to do are just go to the rest of the scripts here and run this. When this is now reorganizing the files that it's just loaded in to fit them in the naming structure and the grouping structure that you've created in your parameter file, which, if you remember, is up at the top here. So it's figuring out which one belongs to which, assigning them, and then reordering the events from those files in one big matrix in order. All right, so we're just going to go through and run all of this. And now you can see here, this is how many events. This is telling us about the data that we've got. These are the files that it's found. If you don't have anything here, it means it hasn't found any files in your data folder. This is the number of samples it has per condition. This should be what you're expecting. This tells you about the flow files that it's finding, what sort of things it's finding. These are the parameter names. That, again, should make sense. This is the total number of events or cells in each condition. And then here you have a summary of what's going on in each individual data file, how many events each one has. You can see if one is contributing far too much. So at this point, you may want to downsample your data. So running, for instance, 50,000 events or by 8 is going to take way too long on your tSNE and isn't really necessary to get a good idea of what's going on. So there's multiple places in the parameter file where you can tell it to downsample. One is for the density parameters. I have not had much success actually downsampling with this, so I tend to leave it as null, which means don't downsample. If we scroll down here, you can see this is where we are going to do the actual downsampling that matters more for the tSNE, the UMAP, and the statistics. So you can downsample or select a limited number of events in 3 different ways. One is in total. So if we told it we only wanted 1,000 events, you put 1,000 here, and it would select those based on how much each one contributes to the total, so proportionally. What we can also do is say, I only want 500 events per condition. So I only want 500 here, 500 here, 500 here, and 500 here. And I don't really care how you get them, so it'll just divide them up proportionally. That might be useful if you had, say, groups that with big mismatches. Say in your knockout, you had hardly any cells, in your wild type, you had a normal number of cells. The one you're most likely to use, the one that I use all the time, is the last one, which is per sample. So you have each of these individual samples, in my case, individual mice, and I want to say, only take, say, 500 or 2,000 cells from each sample. So if I take 2,000 cells and I have 16 samples, I'll have 32,000 events, which is completely fine on just about anybody's computer. You should be able to run probably 100,000 or so. If you have a fast computer, perhaps 200,000. More than that, and you'll probably run into it taking a really long time to complete. You may need to run overnight. And you may also not be able to complete the statistical analysis, the tSNE diff. If you change the number of events that you're going to analyze, you probably also want to change how many iterations you're going to go through, which is down here for the tSNE. So a good number of iterations in general is 5,000. And you can verify this by looking at your tSNE convergence graph that is produced at the end. If you have a lot of events, say over 100,000, you may want to run 10,000 iterations. If you really only have a few events, you might get away with 2,000. It also depends on how slow and frustrating it is to run on your machine. For the UMAP, 1,000 iterations is generally fine. You can play around with that, particularly if you're running this on the cloud with further iterations. All right, so let's stick with this. Now, the script, the analysis script, which is this over here, has already loaded in your parameter file with all of these as variables. So if you change something after starting the analysis, you need to reload that variable. So I have changed this variable from null to 2,000. So then I need to load that into R so that it knows that that value has changed. The way I do that is, again, selecting it and hitting Command Enter. Now, if we go back and run this part again, from the part where we read in the FCS files, we should see that it cuts down to 2,000. where is that? That's down here, actually. That's later. Never mind. Okay, so that's loading in the data. And now let's talk about errors in the next session.